Hello, I'm Renzo, Laurentius, and this video refers to the educational lecture of the 2019 OHBM conference in Rome. And this lecture is a part of a lecture series that is organized by Amir Shmuel. And I want to start by thanking Amir for inviting me in this educational lecture. And Amir asked me to talk about non-bold layer-dependent fMRI methods. So I will do that, and I will first focus on the non-bold layer-dependent fMRI methods that are out there, describe them a bit, how they work, and then in the second half I also want to show you some of my favorite applications of non-bold layer-dependent fMRI, specifically to find something out about the brain, neuroscience applications. Since this is an educational presentation, I will not have the time to really go deep into the details of all the non-bold layer-dependent fMRI methods. If you care about the details, however, I would like to recommend you this review article here. And just in case you're the kind of person that doesn't want to read a lot of text like me and rather enjoy having things explained to you with pretty images, check out this YouTube channel that is called Non-Bold Layer-Dependent fMRI. The title is exactly this exact topic that I'm going to talk about. And in this YouTube channel, I try to collect different videos of other experts in the field where they talk about their point of view of non-bold layer-dependent fMRI. And since it's only 30 minutes, I already now want to apologize if I cannot really show every important contribution. In fact, if you're sitting in the audience and I'm not showing your stuff, please excuse me. This is particularly thinking about the fact that almost everything we know about non-bold layer-dependent fMRI is based on decades of careful animal research. But today I will mostly focus on a human non-bold layer-dependent fMRI. And we heard already earlier this morning why layer-dependent fMRI is so valuable, simply because it can help to bridge the different spatial regimes of neuroscience. From the microscopic side, we have synapses, neurons, and clusters of neurons. And off the macroscopic side, we have behavior, brain areas, and the distributions within. And the keystone is the spatial regime of a few hundred micrometers where we have these layers, and these layers give us a unique window into looking at brain system. For example, we know that feed-forward input comes into a middle layer, whereas feedback input comes in upper and or deeper layers. Or in other areas, we have this input-output model. So layer-dependent information tells us something about the content of the activity. But we also know that we are not really measuring neural activity directly. In fact, we are relying on this neurovascular coupling depicted here by showing firing neurons. We have an astrocyte and the arterial vessel and also a vein here. And as soon as we have activity increase, there's a lot of energy demand of the postsynaptic side, which triggers the arterial vessels to open up. So there's a blood volume increase here, we have a flow increase, and a short while later also on the venous side you see an oxygenation change. And based on this little animation, you can already see two important limitations. The first is that there are a lot of different mechanisms involved, meaning that we are not really sure if the signal is different due to these mechanisms or if the signal is more modulated from the neural activity. So we are not quantitative. Second is that this vein here comes from somewhere, right? So it's not only representing the oxygenation changes of the close by neurons, but in fact, it drains big areas and might also drain neurons that are far away, for example, up to 3 millimeters from different layers. So we are caring about tissue activity, but actually we end up with this whole cascade of different mechanisms. We have vasodilation, oxygen consumption, fluid dynamics here that end up with oxygen oversupply until we get to the susceptibility-induced signal increase. And this is what we measure, and this is what we care about. So it would be much better to avoid all these different mechanisms here and measure one of those first parameters directly, such as directly the arterial vasodilation or the oxygen consumption. And in this way, we would lose a lot of ambiguity of the mechanisms down there. The second big limitations that I showed you on the previous animation is that we have a localization specificity limitation. In the fact that when you just increase the resolution to go from 3 millimeter to 2 millimeter, 1 millimeter, 0.7 millimeter, we still can kind of end up with these fuzzy blobs. And now we just have these blobs at high resolution. And we don't really see the nice stripy patterns that we want to see. And we already know where this comes from previous lectures, namely that most of the oxygenation changes are coming from these larger vessels that are not as nicely laminar aligned like the neurons are. And there are other non-bold layer-dependent fMRI methods out there that are believed to be more locally specific, such as this VASO method, which is my favorite method, which is believed to measure blood volume increase of the small vessels that are closely aligned to the laminar neurostructures. And it's non-invasive, and it's actually pretty simple. The name VASO stands for vascular space occupancy, and it's based on a simple two-compartment model where we have two magnetization vectors here. In this cartoon, a red magnetization arrow represents the blood magnetization and the blue one represents the tissue. And then we start every TR with a global 180-degree inversion pulse that flips around this magnetization, and then they decay with different T1 relaxation times. And since blood decay is slower, there is this exact time point coming up now where blood crosses zero, and this is the time when we play out the excitation pulse and acquire our signal. So right during this time point here, the red arrow was just going through zero, meaning that all the magnetization that we have available at this exact time point is actually only everything that is not blood. So when we consider any voxel that has a lot of volume contributions, we actually only see the extravascular space. The bloods are completely nulled, it's black, it does not contribute to signal. Now when we have activity increase, all the vessels open up and we have reduced signal from the extravascular space. And the signal reduction is then believed to be directly proportional to the blood volume increase. But there are also other non-BOLD layer-dependent fMRI methods out there, such as arterial spin labeling to measure cerebral blood flow. And there you don't really care so much about the amount of blood volume change in your voxel, but you care more about the amount of blood volume delivery along the feeding arteries. So for example, when we care about the delivery to a group of slices like this, we can apply this inversion pulse that flips the spins around from the previous slide only in a specific area, such as down there. And then a short while later, we measure how much of this inverted blood arrived here in our imaging planes. And then we try to quantify the blood flow by subtracting the signal that we get with and without the application of this blood inversion down there. And to give you an idea how these data look like, here I'm showing you an example from Sri, where I'm flipping back and forth between BOLD and VASO activation maps, BOLD and blood flow. And you can already see that for blood flow, we have less activated voxels, right? This could mean two things. It could mean that blood flow measurements are just more locally specific and we have less false positives in our activation map, or it could also mean that we are just noisier and have more false negatives. And in reality, it's usually a mixture of both. And if you average a long time, up to double-digit number of hours, then you can go up to 0.8 millimeter resolution isotropic and get pretty decent layer-dependent CBF profiles out of it. But there's yet another non-BOLD layer-dependent fMRI method, namely Calibrated BOLD, which is used to estimate changes in the cerebral metabolic rate of oxygen, CMRO2. Basically the rate of oxygen consumption. And this works as follows. You just assume that your BOLD signal is a function of a lot of different physiological parameters, such as oxygen consumption and the blood, like the physiology, the vascular physiology, blood volume, blood flow. And you can tease apart these vascular components and oxygen consumption components with a series of different experiments. In the first experiment, the calibration experiment, you give your participants gas mixtures to breathe that have an elevated level of CO2, which means that we change the vascular response without really affecting the neurons and the oxygen consumption. In the second experiment, you do your neuroscientific task, which does result in neural signal change, oxygen consumption, and result also in the vascular response. So for these experiments, for the calibration task, we do have vessel dilation, but we do not have oxygen consumption. Whereas for the neuroscientific task, we have both vessel dilation and oxygen consumption. So when you contrast these two experiments, in this equation, you can tease apart the oxygen consumption for neuroscience experiments. And this is how it looks like in real life data of a finger tapping task. For tapping here, in the motor cortex, we see that for VASO and BOLD, there is signal changes mostly in the hand knob, whereas for hypercapnia, the gas calibration challenge, everything opens up. All the vessels dilate and it gives us some sort of like vascular activity map. And combining all these four measures together in this equation gives us estimates of CMRO2. And it's still kind of like debated and under investigation how well these model assumptions really hold in a layer-dependent submillimeter regime. So let's compare all these different methods to the gold standard, the gradient echo BOLD. And all the most relevant sequence quality features that I could think of are depicted here. We have the sensitivity, basically how well you can detect small signal changes or how long you need to measure, how specific we are to specific layers, how invasive we are, and how available your method is. In other words, if you need to be like good friends with the vendors or have like collaborators that program the sequence for you, or how much in general you need to invest to make it work, how effective your sampling efficiency is, and how quantitative you are, in the sense that in BOLD we are usually only measuring these percent signal changes, but we don't really know percent of what, meaning that we cannot really compare the response across two different areas, across two different people, and so on. And in BOLD, most of the signal changes are coming from the large draining veins, especially with gradient echo BOLD, indicated here as different oxygenation colors for activity and rest. But we have a lot of vessels, so we are super sensitive. We are not very specific. We are non-invasive as it gets. It's super available on every scanner. Sampling efficiency is awesome. But the neural quantifiability is pretty bad because we only have these percent signal changes, but we don't know percent of what. Now let's compare this with ASL. In ASL, we don't have these large draining veins. They are grayed out, but we are in fact only sensitive to the water exchange of the small capillary bed, indicated in the background here as this halo, meaning that we have a much higher specificity. But the availability, like it's getting better, especially since the patent ran out. The sampling efficiency is much lower. You need more experiments with this tagging control, and the quantifiability is also better. Now for calibrated BOLD, we are not really sensitive to these vessels in the first place, or we believe that, but we are only sensitive to the oxygen consumption of the extravascular tissue, indicated here as the different gray values in the background. And that method really shines on the neural quantifiability because you can actually measure the oxygen consumption. However, all the other parameters are not the best. The specificity is only as good as the model. It's not really non-invasive anymore because you need to give your participants these gas mixtures, which can be quite unpleasant. And also it means that it's not so available because you need to have this extra hardware component, this gas delivery system, some safety measures in there to make sure that the concentrations are right. The quantifiability, however, is awesome. Now the VASO, the blood volume measure method, which is my favorite, it's not really the best on any of those quality measures, but it's also not the worst on any of those. It gives us, I think, a good compromise for all of them. So in conclusion, I think that any non-BOLD layer-dependent fMRI method is not as sensitive as gradient echo BOLD. And now it's just a question if this is wanted or unwanted sensitivity loss, because we don't want to be sensitive to these unwanted large draining veins, right? All non-BOLD layer-dependent fMRI methods are more specific and more quantitative. And until now, I showed you a lot of different non-BOLD layer-dependent fMRI methods out there, but actually there are many, many more, almost impossible to list. And I even only focused on the ones that are dependent on vascular responses. But there are also non-vascular ones, such as it has been shown that with dynamic measurements of functional diffusion fMRI, you can quite well separate the response in the middle layers without these descending vein biases. Also, it has been shown that with dynamic measures of T1 rho, there are non-vascular changes of CSF volume, especially above the upper layers, which we need to keep in mind when interpreting our signals. So I have about 15 minutes left now, and I would like to use them to show you my favorite applications of non-BOLD layer-dependent fMRI. And these are mostly things done in Peter Bandettini's group at NIH, and also some studies together with Emily Finn. And the first study that was done in this setup was a kind of validation study in the motor cortex, where we actually already know what to expect with respect to layer-dependent activity from animal tracer studies. Namely, we know that most of the cortical input is combined in the superficial layers of the motor cortex, close to CSF, whereas the cortical spinal output mostly emerges from these loops in the deeper layers. And we can try to manipulate these different loops with different movement and touching tasks. So for example, for a tapping with touch, we move the muscles, right? So we definitely have this cortical spinal output down there, but we also have this touch component, which probably results in sensory feedback into the motor cortex in the superficial layers. We also have like motor planning input, which results in a double response. Mainly we have this feedback response up there and also the output response down there. Visible also here as these two independent peaks in layer profiles. Now for the other condition, for touching, we asked the participants not to move their hand, and they really didn't. It actually seems to be that there's a suppression of output in the deeper layers, but still we did this touching, meaning that there's sensory feedback into the motor cortex in the superficial layers visible up here. And this made us quite happy that we can like differentially activate upper and deeper layers, and they're not just scaled versions from each other. However, these results also taught us that you cannot really do layer-dependent fMRI without knowing where you are in the columnar domain too. In the sense that in the motor cortex we have these tiny patches, and going through slices and looking at noisy data, you can kind of always pick a patch that kind of matches your hypotheses, right? And as soon as you are a bit off, like just a few millimeters, then it completely dies away. So we need to know where we are. And in order to develop this kind of topographical mapping simultaneous to layer-dependent fMRI, we also use this kind of finger tapping task in order to get towards the columnar dimension. And to show you how this works, I would like to show you this little animation here where I'm starting with an axial-like slice where on the left hand we have the sensory cortex and on the right hand we have the motor cortex. And you see the motor cortex is much, much thicker. And in order to get towards this columnar representation, we use a crawling algorithm directly in voxel space of the EPI data that kind of grows these surfaces for sensory cortex here in black and for motor cortex here in white or grayish. And this really helps us to have these surfaces in voxel space so we can look at the slices and the surfaces in the same space, in the same analysis, and now basically rotating it to look at the entire central sulcus, now at the posterior side, the sensory side, where the midline would be here and the right ear would be here. And then we can project the individual fingers onto it. This would be the pinky finger, the ring finger, the middle finger, the index finger, and the thumb. And you can see that in the sensory cortex it's pretty like somatotopically organized, in the sense that neighboring fingers are located neighboring in the cortex, and it's pretty big, the representation. Now more interesting is what happens on the motor side. When we flip this around and look at it here in this kind of cave of the motor cortex, where again we have the pinky finger, 2 ring fingers, 2 middle fingers, and 2 index fingers and 2 thumbs. So in the motor cortex it's kind of different than the sensory cortex. We have multiple representations and it's kind of much smaller. So especially this hand representation here is only covering about 4 millimeters. So it's very hard to see in conventional BOLD, and maybe better visible also with respect to the size when we look at this like slide cut like this, where we have the sensory cortex on the bottom left and the motor cortex on the top right. And there you can really see how small these finger representations are in the motor cortex compared to the sensory cortex. And also, again, you see how the non-BOLD layer-dependent fMRI can help us here with the higher specificity, because in BOLD, even though there are similar features visible, you're like as soon as you have one vein kind of going through your cortex, then the whole 2 millimeters around it are dominated by this. And this kind of mirror pattern is also very consistent across participants and also matches pretty perfectly with patches in the motor cortex that are preferentially activated for grasping versus retraction tasks. So the motor cortex seems to be much more like action map representation than a body part representation. And everything I showed you until now really referred to the motor cortex because it's so easy. And people told me that doing layer-dependent fMRI in order to understand how the brain works is like trying to understand how a truck engine works by looking at it with an infrared camera. You only roughly know what's going on and where energy is being consumed, but you don't really learn a lot about the content. For example, if this heat is like where the truck was going, or if this heat is coming from input or output activity. Similarly for fMRI in the motor cortex, where we don't know much about the content and beyond that the motor cortex is involved. Only with layer-dependent fMRI we are approaching this regime where we can say something about the densely packed microcircuitry, for example counting the stripes. If we have 2 stripes, it's probably tapping. If it's 1 stripe, it's touching. But the motor cortex is easy. It's like not like any regular truck engine, but it's like the biggest engine there is. There's no cortex as thick as the motor cortex, and you don't need any fancy methodology to easily see the densely packed microcircuitry of an engine of that size, right? You see input and output even with a small field of view. And this is the reason why we invested quite a lot of time in the recent years to extend this layer-dependent fMRI methodology for non-bold contrast, also for more regular-sized cortices, such as the visual system. And we wanted to get there without really sacrificing this stripiness. For example, here these dark voxels forming a line, indicating the tree of Gennari, and we want to get there covering an entire slice, not only with a small field of view, also part of an entire slab of isotropic voxels. And since we care about activity and not only pretty EPIs, we need more than just the blobby bold, but actually we need a higher specificity of non-bold, VASO layer-dependent fMRI, even though it comes along with a higher noise level. And to see if this is also applicable, we follow Jonathan Polimeni's work here, which will be the next speaker, and to look at direction connectivity in resting state between the thalamus, V1 and V5, which have different feed-forward and feedback connections, respectively. Here, for functional connectivity within V1, we see that for a seed region in the thalamus, we mostly have this feed-forward peak in the middle layers, whereas for a seed region in V5, we mostly have this feedback and peak up there too. And this made us confident that we can actually apply this in neuroscience, and we tried to kind of leave this very well-controlled regime of these test cases where you zoom into a patch of cortex and then only investigate these differential responses for different seed regions or different tasks. Why not do it the other way around and look at one task condition and look at differential responses across the columnar domain? And again, here we are assuming a canonical microcircuit, but we just assume that a feed-forward driven column would look like this and a feedback driven column would look like that. And then we can use resting state data and just pick a seed region and see what's feed-forward or feedback-driven. For example, here picking a seed region in a cluster of voxels that cover LGN and the pulvinar, you see that most of the visual stream is red. It means that they receive feed-forward input. But when we move somewhere here between V1 and V2, you see that maybe the calcrine sulcus actually receives quite a bit of feedback too. The higher order areas, still mostly red, receive feed-forward input. And you can move along here to the next step or the next here, which might be V5, where most of the early visual areas really receive feedback input from the seed, and only these further anterior areas still receive feed-forward input. And in this way, you can kind of map out the cortical hierarchy on a column-by-column domain without predefined ROIs. Another application for non-bold layer-dependent fMRI in a more cognitive neuroscience domain is this study that has been conducted by Emily Finn, and she will present this on Wednesday. And also Martin Havlicek has some simulations of layer-dependent fMRI with respect to this study, I think also on Wednesday. And there we looked mostly at the dorsolateral prefrontal cortex, which has very specific functional canonical microcircuits, in the sense that there are different cells. These black cells are cue cells, we have delay cells, we have response cells that eventually trigger the motor system. And they are all located across different cortical laminae. And Emily Finn found this task that should engage these different cells differently. For example, by showing participants these strings of letters and then cueing them to either memorize these letters in the order they saw them or sort these letters according to an alphabetical order in their mind, which is very hard in this believed to engage the superficial layers. Later in the task, we show them random selected letters and they need to do a response decision, which then expects us to believe that there's activity in the output layers, the deeper ones. And this kind of simplified canonical microcircuit really nicely explains the data that we are getting. Here, for example, zooming into a CBV-weighted EPI that has this inherent T1 contrast, where the response condition really nicely follows the deeper layers along the cortical ribbon here upon a smoothing of 1 millimeter within layers, whereas the alphabetization condition really nicely a parallel line of the superficial layers, made better visible even here in the group results of the time courses of 16 participants averaged, where the working memory superficial layers, they shoot up as soon as the participants start with this mental alphabetization, and when they found the right order, it goes back to baseline. Whereas the deeper output layers, they don't really care so much about this alphabetization, and they only shoot up as soon as they need to do the response much later. And if you want to hear more about it, check out Emily's preprint here. Another realm of cognitive neuroscience that's really underdeveloped for non-bold layer-dependent fMRI might be these naturalistic tasks like movie watching. And they might actually be very well suited for non-bold layer-dependent fMRI because you can use them to test the ambiguity of connectivity studies without this like uncertainty if you have like physiological noise or neural driven fluctuations. And we started to approach this new task regime by using the same movies that are also used for the 70 HCP data release, where we know already that we have 100 participants, we know which area is supposed to react to these movies, and we use the same movies for high resolution VASO data. And again, the VASO can help us here really nicely to distinguish between physiological noise that's only visible in BOLD compared to neural-driven fluctuations. Here, for example, you see that in the default mode network, we have all these like false positives where the large vessels are, and with VASO together, we can kind of remove these physiological component. And the kind of new questions that we might be able to address with this setup refer to to better understand the sources of individual differences. Here, for example, we just pick any network like the default mode network, and we know from the HCP data that in this network, most people really have very similar signal traces. However, there are still time frames that are more stable across people compared to other time frames. In fact, when you show people these like beach scenes and wave patterns, almost the entire brain does the same thing no matter what you do. Whereas when you show them kissing scenes, people tend to react more differently. And then we can zoom into these time frames and to those scenes and see where these differences are coming from. Here, for example, visible that in this case, it really seems to be from the feedback-driven layers, the superficial ones. Whereas the deeper layers, they are very consistent across people, which I think can make a lot of sense, right? Because these like feed-forward input must be the same, right? All the people look at the same movies, so the like activity of the retina must be the same, or the activity of the early visual areas extracting the low-level features must be the same. Only somewhere along the cortical hierarchy where these individual differences are coming in, these differences must come in at feedback input. Other questions that we might be able to address with this setup refer to the like hubness or direction connectivity of already described networks, such as the default mode network, where we can use the same setup. We just extract the signal time courses of the averaged HCP data. So we know that the default mode network should look like that. And we can use these signal time courses as regressors or as our design matrix in our high resolution data while people watch the same movies to extract the default mode network and also give us a layer response. And these layer responses then might be able to help us with respect to the hubness. So for example, you see that the IPC or PFC are really feedback driven, very different to the PCC, which does not have this strong feedback activity up there. And these kind of layer profiles might be used in reasoning to say which area is the hub of the network. For example, suggesting that some areas might be just passively being dragged along with the hub, whereas the hub is only the driving force. Other things that you might be able to do with non-BOLD layer-dependent fMRI is a study here conducted in the primary sensory cortex with respect to predictive coding. So in this primary sensory cortex, we expect very similar canonical microcircuits like in the default mode network, like in the primary sensory areas, in the sense that thalamic input mostly is expected to be coming in the middle layers, whereas the cortical feedback is mostly coming from upper and a deeper input. And we can try to engage these different channels differently by giving people different touching sequences. So participants learn specific sequences of being touched, and they're always asked to predict which finger is going to be touched next. And here they are touched in a certain order and then expect to be touched next on the index finger. In some instances, however, we left out the index finger. And in those cases, they expected to be touched there, but there was actually no feed-forward input happening from the green side, only expectation from the blue side. And in one of the many control conditions, the participants were touched in a completely random fashion. So here, the people could not predict which finger is going to be touched next, but there was actually feed-forward input happening from the middle layers here. And for these two cases, we pretty much get what we expected, namely that for feed-forward condition, we mostly have the middle layer activation in this small patch of the sensory of the finger representation in the primary sensory cortex, whereas for the expectation predictive coding condition, it's mostly the superficial layer. And this might be again better visible also in the group results of the profiles, where we see a clear peak in the blood volume response in the superficial layers, whereas for the feed-forward condition, it's mostly a peak in the middle layers. And with this, I thank all my colleagues and I thank you for your attention.